So just as we were talking about previously, the quote from the Nature paper by Watson and Crick, where they speculated that the double-stranded complementary piece of DNA was serving basically as a great template for the synthesis of new DNA, that's actually what we observed, or we observe. And so essentially, one strand of double-stranded DNA serves as a perfect template to produce basically identical copies of that DNA. And this synthesis is catalyzed by enzymes known as DNA polymerases, which when you think about the name there, DNA, okay, so that could be your template, or that's what you're synthesizing, you're producing. It's making a polymer, and this ASE suffix, we're going to see that more and more whenever we talk about enzymes. So a newly made DNA molecule has one daughter strand and one parent strand. So one from the original and then one that is a newly synthesized strand. Now, messenger RNA, mRNA is what it's also known as, is the code carrier for the sequence of proteins. This is synthesized using DNA as a template and generally occurs as a single strand. This is not always the case, but more times than not, it is a single strand. What makes it kind of unique is that it can fold in on itself and make a double-stranded RNA, but that just has to do with the possibility for any sort of hydrogen bonding between base pairs. So think about it as a single strand that floats around. However, that single strand can fold back in on itself, but more times than not, it's going to exist as that single floating strand. What's important to recognize about mRNA is that it utilizes the sugar ribose rather than deoxyribose, and in place of thymine, it utilizes the nitrogenous base uracil. So one mRNA strand may code for more than one protein. This is known as a polycistronic piece of mRNA. And ultimately, together with the transfer RNA, or tRNA, this is going to transfer genetic information from DNA to a protein. So the central dogma of molecular biology is DNA to RNA to protein. Now, the reason that we kind of leave this as RNA is because, well, mRNA contributes, as does tRNA. So what I was saying a moment ago about an mRNA strand having or harboring more than one gene, well, that's kind of what we're looking at here. We have a monocistronic gene, or monocistronic piece of mRNA versus a polycistronic. As you can see, the polycistron has 1, 2, 3 genes within it. Now, palindromic sequences can form hairpins and cruciforms. So a palindrome, for instance, whenever we think about numbers, a palindrome would be like 101. It's a word or a phrase or a number that is the same when read backward and forward. Here we got some examples. Rotator. For some reason, it feels like saying it like that. This word, which is apparently Finnish for soap cup batch trader, which that sequence of words doesn't really make any sense to me in English. I don't know what a soap cup is. A cup for holding soap, or is it a cup made out of soap? I'm unsure. This sequence of characters, which I believe that would be the Cyrillic alphabet. I'm not sure exactly. So at any rate, let's go with civic and 101 as really good examples of palindromes. Now here what we have are examples of palindromic sequences on either the same strand or in different strands. If you look at this one up at the top, what we've got is reading from the 5' to the 3' direction on this strand. You see pt3a, GCAC, and so on. And then if you look at this bottom strand, or this opposite strand, reading in the 5' to 3' direction, it basically has the exact same sequence. Now this minor repeat shows that we've got a palindromic sequence on a single strand. The implications of this are of importance because ultimately what can happen is what I was talking about a little while ago about an mRNA basically folding back in on itself. Here we have a strand or a piece of DNA, as it would be. I know this is DNA because I see that T there. Um, what can happen is this single strand can find a complementary region within itself and fold and make this hairpin sequence. So we've got this hairpin region right here, which ultimately is kind of not desirable because this wouldn't fold up into a double helix, and so it wouldn't be, I don't know if I want to say it wouldn't be retained, but it has internal forces that are going to cause that one strand to basically loop back on itself. Now, when it comes to RNA molecules, this is something that you see from time to time. And we see some of these structures that are pretty complicated. So this whole thing right here is one strand. And it's kind of tough to see, but right here and right here, we've got our 5' end and our 3' end of this whole sequence. So it's actually kind of like a maze where you can follow it and you can look at the numbers of those specific nucleotides. But as you can see, there's lots of regions of this piece of mRNA that have complementary regions within itself. And sometimes those are going to result in formation of these hairpins. So here, 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 these are all hairpin loops and hairpin structures that, well, there are just attractive enough forces between those, or hydrogen bonding that takes place between those individual base pairs. And so you can, I don't know if I want to say you can grow this structure like this, but a structure as kind of massive like this can ultimately develop. Now, these complex structures are stabilized by our non-Watson-Crick base pair interactions. So examples of those are interactions that, they're not A, U, I'm saying U because we're talking about an RNA molecule, and they're not G, C, but instead you have things that are taking place that are a little bit atypical. For instance, here is a cytosine guanine, so a GC bonding, but then you have the 7-methylguanine. That 7-methylguanine is a guanine residue that has been methylated at position number 7, which is right here. Now, what can happen there is the hydrogen bonds can form between this 7-methylguanine and a cytosine and a guanine simultaneously. And that's stabilized ultimately by a negative charge on the phosphodiester backbone. Okay. In addition to that, you can have adenine N2-dimethylguanine and adenine. What's hidden behind my head here is another adenine. So we've got this 3-base pairing between adenine, a uracil, and another adenine nucleotide. So these complicated structures, they're not necessarily always going to be spontaneous, but they can be basically, the complexity of them can be enhanced because of these non-Watson-Crick bases, or these non-Watson-Crick base pairing taking place with these modified nucleotide derivatives. So the next thing that I'd like to talk about is basically the stability of DNA. So think about a DNA molecule and what's inside of it. We've got negative charges on the exterior, then we've got hydrogen bonds between our base pairs. So those hydrogen bonds, well, when you think about the strength of different types of bonds, a hydrogen bond versus like a covalent bond or an ionic bond, well, a hydrogen bond is not extremely strong. However, in numbers, they basically develop strength. It's kind of like the ants in A Bug's Life. Nobody really liked A Bug's Life, but I always loved it. They have strength. An individual one is not particularly strong, but in mass, they're very strong. So whenever it comes to a DNA molecule, well, we have this process of denaturation, heating it to separate it. So this is not exclusively heat-based, but this denaturation can take place by the addition of a high temperature or a change in the pH. So that denaturation can be undone by annealing those pieces of DNA. So we have this denaturation, or I sometimes refer to it as melting. We have annealing that brings them back together. So the annealing is the undoing of denaturation. Now, this partially denatured piece of DNA ultimately can be continued ultimately till you get two separate strands of DNA. So these hydrogen bonds within the interior of that molecule, those are going to be broken. Now, what that is going to mean is that the base stacking that you had taking place is going to be lost, which is going to mean you have an increase in UV absorbance, which, keep in mind, we had 250 to 270 nanometers as the wavelength of light that is absorbed by these molecules. So think about it as the UV absorbance is increasing, and if the UV absorbance is an indicator of the concentration per the Lambert-Beer law, or Beer's law, effectively it's like you've got one molecule of a nucleic acid. When you separate them, you have two. So the concentration appears to go up because your absorbance has gone up. Now, that thermal denaturation, or melting, is a very important part of the polymerase chain reaction, also known as PCR. PCR was devised or developed by, or is credited to a guy named Kary Mullis. He recently passed away. I think it was in the past 3 years or so. But DNA exists as double helix at room temperature, and it's very, very stable, which we previously talked about. These two DNA strands are going to dissociate from one another at high temperatures. Now, these two strands will reanneal when the temperature is lowered. This DNA denaturation is commonly monitored by UV spectrophotometry at 260 nanometers. So you can see a change in absorbance at 260 based on this melting. Now, what that ultimately is going to look like is something like this. So this is a DNA melting curve. On our axes, we have denaturation, or percent denaturation, and on our x-axis, we have temperature. So as our temperature increases, well, we see our midpoint here change. So what I'd like to draw your attention to first is this Tm for our first molecule. And in blue, that's represented that 50% of our molecule has been denatured at, let's call that 83 degrees Celsius. The next molecule, it takes an increase in temperature up to 85 degrees to get to that 50% denaturation. At upwards of 90 degrees, both of these molecules are effectively 100% denatured. But this is a denaturation curve right here. And specifically, this is a thermal denaturation curve. Now, what caused that shift of our two lines? So we had one like this and then one like this. So 1 versus 2. What's different about those two molecules that caused that shift to the right? Well, like I said, the midpoint of the melting temperature depends on the base composition. So if you have a higher percentage of GCs, or G's and C's, within that molecule, you have an increase in, well, hydrogen bonds, because each one of those G's and C's has 3 hydrogen bonds between them. Now this Tm, or this melting temperature, is going to depend on the DNA length. So a shorter piece of DNA will have a lower melting temperature than a longer piece of DNA. Now, the melting temperature is also going to depend on the pH, because hydrogen bonds are going to be influenced by the pH of the solution, as well as ionic strength. So if you increase the salt concentration, your Tm is also going to increase. So we see this shift from left to right. Now, this would be a curve that shows the correlation between GC percentage and melting temperature. And so what we have here is, as our GC content, or our percent G's and C's within the total molecule, as that goes up, we have an increase in the Tm. So keep in mind that Tm was the temperature where 50% of the molecule was denatured. So with all of that said, when you think about a enormous DNA molecule, the melting is going to have, or it's going to be influenced by what the composition of that region is. Is it a high AT rich or is it a high GC rich? What this is showing is in a very large piece of DNA, we've got regions that are highlighted in red, here, here, and here. Well, there are many others, but what we're seeing there is that we're seeing the separation of our two molecules, of our two strands. And what that is indicative of is a high AT region, whereas much of the rest of this, we've got a high GC content. GC, I don't know why I put that. A high GC content, or at least a kind of equal peppering of both A's and T's and G's and C's. So it's not a particularly high AT or a high GC content, but it's kind of a region that's evenly distributed.